Hello, everybody. Welcome to Creation Myths, or welcome back to Creation Myths if this is not your first time here. Welcome back. We have some fun stuff to talk about today. So let me give you the rundown of what we're doing here. Today's video kind of starts with a creation myth that I published a couple weeks back. It was the creation myth on the claim from creationists that any biological information requires an intelligent source. Right? That's the creation myth. Now, that's not true, but that's the claim. That's what I did my video on. So I based my video on a couple specific things, but one of them was a video from Dr. Stephen Meyer from Discovery Institute. And in that video, Dr. Meyer said, and this is a direct quote, he said, information, especially in a digital form, always comes from an intelligent source. Right. And that's kind of a good summary of the claim that I was addressing, that you need an intelligent source, God, a designer, whatever, in order to get biological information. And I showed that's not the case by referencing a couple of experiments that involved using random sequences to generate some kind of biological function. You generate random sequences, put them in an organism, and show that there's either some biological activity that happens, or in one case, there is a particular function in one of those experiments that they were looking for, and they were actually able to document that function coming from randomly generated sequences, right? So Dr. Meyer makes this extremely specific claim, and I refute it with a couple of specific experimental references, right? So what I was just delighted to see this afternoon was this article over on Evolution News. That's kind of the blog from Discovery Institute. So they published a piece by Dr. Brian Miller unraveling the myth that undesigned processes generate novel functions. And I saw that and thought, gee, that's interesting. I just talked about that a couple weeks ago. I'm going to read this. I bet this is going to be really interesting. So let me come down here. And I was recently informed of a video posted on the YouTube channel Creation Myths. And aha, that's why that sounded familiar, because this article is about the video that I put up. Now, before we go, I'm just going to go down to the bottom and just highlight this author, because I am not familiar with Brian Miller, but we come down here all the way to the bottom. This is not long, thankfully. It's only about 4 pages, so this won't take us too long. I always say, and then it takes an hour, right? But we'll see how long this takes. So Brian Miller, he's got a BS in physics, minor in engineering. Always engineering, right? It's always engineering. It's that Salem hypothesis again. PhD in physics from Duke. Okay, so he's got legit credentials, not bio, but like he's got a legit PhD. So what we're going to do here is we are going to go through this response from Dr. Miller and see how they address my specific claims, or, if they address my specific claims. We're not going to read the entire thing word for word. Like I said, it's not long, but we're not going to read the entire thing. We're not going to sit here and go word for word. We're just going to go through. You can see as I read it, I highlighted, and then I underlined some particular things in red that I want to make sure that I remember to talk about, right? So that's what we're going to do. So we're just going to go through this top to bottom and see. So this is just, if you found the video, Creation Myths, there's a link to it. It's a nice one to link it. The video specifically references a clip of Dr. Meyer. Meyer states that the information required for the sudden appearance of radically new animals could only have originated from a mind. Now, he does say that, but that wasn't the claim I was addressing, right? So right away, we're kind of not exactly on target in terms of what I was talking about, because I wasn't talking about the Cambrian explosion. I wasn't talking about new animal forms. I was just talking about digital information. The specific quote that I used was Stephen Meyer specifically referencing digital information and giving a number of examples. And I'm going to give you a longer version of that quote in a little bit and show you the examples he means to show you that it's exactly relevant to what I was talking about, right? So to continue, he says the critic responds to the claim by arguing that experiments have demonstrated that information can be created by natural processes, and he cites 2 research studies to support this assertion. Yes, that's basically what I said. Fair summary. Thank you. His argument ultimately fails since it is founded on a misunderstanding of the evidence for design associated with biological information. This error is so common, it deserves special attention. All right, so we're going to go through, we're going to do this. So he kind of goes through and summarizes each of the two studies I talk about, and then talks about why my my kind of assertion based on those studies is wrong. My interpretation of those studies is wrong. So first was the Nimi et al. 2017. This is the random RNAs and polypeptides are an abundant source of bioactive, or random sequences are an abundant source of bioactive RNAs and peptides. These papers, by the way, they're referenced in the original video, which is linked down below. So if you click down below, go to the original video, the links to these papers are in that video description, in addition to the link to the original Stephen Meyer video that I was referencing. So in this study, what they did is they randomly generated 150 base pair sequences. They stuck them in some E. coli with a plasmid and found that about a quarter of them were beneficial to the E. coli. And then in the second article, that's Yona et al. 2018, this is the random generation of the E. coli lac promoter. So what they did here is they found, they used a string of 100 random nucleotides, 100-ish random nucleotides, and found that about 10% of random sequences adjacent to the genes in a lac operon would bind to the polymerase in such a way to initiate transcription, which is a roundabout way of saying that 10% of random sequences functioned as a lac promoter. That's what he says here without saying the words. 10% of random sequences functioned as a lac promoter. Also worth noting that within one mutation of a bunch of those random sequences, within one mutation, most of them function as a lac promoter. Some of them actually better than the original lac promoter that they replaced. So this is underselling that experiment a little bit, but it's not wrong. It's just, I think, underselling it. So let's continue. He gets into, what's wrong about these two experiments, why these things don't count, basically. And he's going to start with the Nimi et al. Paper, the 2017 one, and quotes from Douglas Axe writing about this at length. And this is a little out there because I don't really know what Axe is trying to say here. I'm going to read it because this is something else. He says, they, the authors, Nimi et al., merely showed that if you burden bacteria by forcing them to churn out RNA and protein from random inserts, it's fairly easy to find sequence-dependent effects on growth. Not because anything clever has been invented, but because the burden of making so much junk varies slightly with the kind of junk. That means any junk that slows the process of making more junk by gumming up the works a bit would provide a selective benefit. Such sequences are good only in this highly artificial context, much as shoving a stick into an electric fan is good if you need to stop the blades in a hurry. And then the author of this piece says, in short, the sequences perform no new function, so no new information was created. Now, I don't think that's a fair representation of that paper at all. The authors, first of all, didn't claim new function. So right here, no new function, therefore no new information. We'll get to actually, let me just say, who says you need a new function for new information? That's not the standard. Okay. You don't need a new function for new information. That's not the state, right? That's new. But putting that aside, the authors didn't claim they were functional. They said they were bioactive is the word they used. In other words, they have some kind of chemical activity, right? Whatever the activities were, and we don't know what all the activities were, but whatever the activities were, some were beneficial to those bacteria, and many were not. Now, that means, and the authors confirmed this, that the effects of those random insertions, what they did is they took a plasmid, little circular piece of DNA with this random insertion in it that they could express, and they put that in the bacteria to express that little bit, either as RNA or as a peptide, if it had a start codon somewhere in it. So what they found were that the effects were sequence dependent. right? They don't know what those effects are, but they found that those effects were sequence dependent. And they focused in more detail on 3 of the sequences they tried out. They tried like 100 something. And they focused in detail on 3. And really interestingly, what they found was that 2 of them were active as RNAs. They were not transcribed. They were not translated. They were transcribed. And the RNAs, the specific RNA sequences, exerted some beneficial effect. And the third was actually active as a polypeptide. So it was transcribed and translated, and that short polypeptide did something that was beneficial. There's no basis for claiming that they're junk, as Axe does here. He's just asserting without evidence that, like, oh, they didn't do anything. He's basically saying this takes the attention of the transcription and translation machinery that would otherwise be focused on doing other stuff, and that other stuff is more costly than the stuff they inserted. Therefore, expressing the stuff they inserted had a net beneficial effect. There's no evidence that that's actually what happened. And in fact, the sequence dependence indicates that that's probably not the case. There is probably some specific activity here that's sequence dependent and transcription or translation dependent, depending on which insert you're looking at. That means that what Axe and the author here are claiming is that there is a novel, specific sequence having a positive effect on growth through some biochemical means, but that isn't new information. Right? That's the implication of what we're talking about here, that those series of things don't count as new information. Okay, fine if you want to make that claim, but I don't think anyone's going to take that seriously outside of creationist circles. It's sequence specific. It has some biochemical activity that has a net beneficial effect on the cell. It's either functioning as an RNA or a peptide. And they only, again, surveyed 3 of those 100-something sequences in detail, not all 25 that had a beneficial effect. I bet they'll find more active peptides if they surveyed the rest of them, or such as 25% that have a beneficial effect. I bet they find more polypeptides that are beneficial if they do that, right? But that doesn't count as information. Okay. So let's go on to the second paper, Yona et al. So this is, I love this one. This is about the E. coli lac promoter. I think this is really cool. So here's what we get written about this. The Yona et al. Experiment did show that a DNA sequence can be randomly generated that can perform simple functions such as binding a polymerase. Great. Earlier, they said it didn't do a function. Therefore, it wasn't new information. Boom. Random sequence performs a function. New information. Done. We can all go home, right? According to the standard that they just established like 5 minutes ago, this is it. Then we're good. We could ignore the first paper where the second one covers it. The second one invalidates the creationist claim, right? Of course not. We'll find a reason it doesn't count. So let's continue. Meyer is not claiming that random processes cannot generate small quantities of information. He is arguing that random processes cannot generate the quantity of information required for anything comparable to creating a new protein with a novel structure. I'm going to throw a flag on the play, moving the goalposts for this one. It's a pretty extreme goalpost move, I think. I promised earlier I would give you the longer version of what Meyer said in that video. Here's Dr. Meyer's exact words, the longer version that I didn't originally quote in the original video. Now remember, what we get right here from it's Dr. Miller, right? It's Dr. Miller up here. It's Brian Miller. What we get from Dr. Brian Miller is this. Meyer is not claiming that random processes cannot generate small quantities of information. He is arguing that random processes cannot generate the quantity of information required for anything comparable to creating a new protein with a novel structure. Here is the longer quote from Dr. Meyer. Information, especially in a digital form, always comes from an intelligent source. Whether we're talking about a paragraph in a book, or a section of software, or a hieroglyphic inscription, or even information embedded in a radio signal, whenever we see information, and we trace it back to its ultimate source, we inevitably find a mind, not a material process. Does it sound like Meyer was limiting his claim to a specific level of complexity or a specific quantity of information? It does not seem like he's doing that. I want to emphasize those specific things, a paragraph in a book, hieroglyphics, a radio signal, whatever, those are exactly the kinds of information that I address in my video. We're talking essentially about sequence information, right? A series of something in a row that conveys a message, whether it's letters on a page, nucleotides in a genetic sequence, right? That's exactly the kind of information we're talking about. Meyer is not setting a threshold here. Well, you need an intelligent source to get a certain quantity of information, right? So let's look at two problems here. Two problems associated with this, the quantities of information and then the new protein with a novel structure. But there's two problems here. First, how are we quantifying the information to make this claim? Because he is arguing that random process cannot generate the quantity of information required. Okay, so what is that specific quantity? How are you measuring it? How are you quantifying that information? Once we have answers to those questions, we can ask, what's the quantity of information that is generated in the two papers I referenced? What is the quantity of information using the same methodology to quantify it in those randomly generated lac promoters? Do we have any of that information? Did anybody at any point attempt to quantify any of this and make those comparisons and show, oh, well, you can generate this much. This is how much is in those lac promoters, but you get up here and that's just too much information. You can't get there, right? No, nobody's doing any of that. This is just an assertion made without evidence. Okay, so that's the first problem. We're invoking a quantity threshold, but we're not doing anything to support that assertion. We're not giving any data. We're not giving any limiting principle. We're just asserting that there is somewhere a threshold quantity of information. And like, yeah, sure, you get below it. That's fine. But you can't get to that threshold, which would be making a new protein with a new structure would be beyond that. Okay. Second problem here is with that phrase, new protein with a novel structure. Let's think for a minute about how we get new stuff in our genes. How do we get new proteins? How do we get new genes? There's a lot of processes in there, and requiring that we get a new protein with a novel structure, which I interpret as meaning it can't be based on any pre-existing thing. Think about what that excludes. No gene duplication followed by neofunctionalization. No pseudogenes and refunctionalization. No gain of function mutations in existing proteins that confer new functions. No horizontal gene transfer of any kind. Because even if it does something new in the recipient, it came from somewhere else. It's not a new structure, right? It's all based on something pre-existing. What they're asking for is kind of the same thing that like Dr. Michael Behe does when he is very particular when he talks about like Darwinian processes to generate irreducibly complex structures. It's a little bit tough to pin him down on like what processes count. What are you including and what are you excluding? Same thing is going on here. By saying new protein with a novel structure, they're limiting it to de novo proteins that come from not just non-coding RNAs, but like just total nonsense DNA in the first place, which is a rare thing that happens. It happens, but it's a rare thing. And it excludes most instances of new protein coding genes that we know of. We're just excluding the processes that actually generate the results they're looking for. So we have lots of documented examples of evolutionary processes generating new, information, however they want to define and quantify that, but it's being excluded, I think, with this framing of the question. But let's take their argument at face value anyway. Let's just limit it to de novo proteins. Does their argument hold up? No, it does not. And to show you a few examples of that, I'm going to switch the screen. So we're going to take this down, and hopefully I do this correctly. Now let me share just a quick other little thing here. This will be a very small thing, and then we'll go back. It's this right here. I just want to show you a smattering of the papers that are addressing this question, the question of de novo genes. So this is, again, this is not all of them. This is just some of them, but I want to share them with you. So here we have de novo gene evolution of antifreeze glycoproteins in codfish revealed by whole genome sequence data. These are a whole bunch of de novo protein coding genes in fish that live in extremely cold water, and it's essentially antifreeze genes so that they can survive in the extremely frigid ocean. There's been a bunch of follow-ups on this about like the actual genetic makeups of it. It's really cool. Then we have rapid evolution of protein diversity by de novo origination in Oryza. So we have a whole bunch of new protein coding genes there. Then we have, but this is so we got fish, we've got plants, then we've got origin of de novo genes in human and chimpanzee from PLOS Genetics, and related to that, hominoid-specific de novo protein-coding genes originating from long non-coding RNAs. This one is really cool, I think, because it provides a mechanism. Just to take a minute to tell you about how they found these de novo genes, what you're looking at is in one lineage, say humans. We have a bunch of protein coding genes that chimps don't have. It's like, oh, that's weird. How did that happen? Well, we know. What you look for is where you have the gene in the human that doesn't have the same protein coding gene in the chimp genome, but what you can often find in the chimp and the gorilla and other apes are that sequence, but it's transcribed, non-translated. So it makes what's called a long non-coding RNA. Now, those RNAs often don't do anything. So like in the chimp genome, it maybe makes that RNA, it's very short-lived, it gets broken down. In the human genome, you have a mutation that confers a start codon. Now it can be translated. So that long non-coding RNA in one genome becomes a protein coding gene in a related species. We now know how to find these things. We know what to look for. And we find them all over the place. So we now have a mechanism that we can test and say, okay, prediction. If de novo protein coding genes tend to come from long non-coding RNAs, let's go look for those. Let's go look for those out in related genomes, and then boom, we find them. Okay, so even if you go by the standard that they're saying, then we have the answer to the question. We have examples. And I'll cite one more, and I love this one. This one's in yeast, which is cool. So this is de novo emergence of adaptive membrane proteins from thymine-rich genomic sequences. This one is really cool because this one was actually in the lab. So they demonstrated not just experimentally that new genes could form, but they actually show the mechanism and how, once it became protein coding and actually expressed as a protein encoding gene rather than just a non-coding RNA, once you had that expression, they showed how it could be optimized via selection, right? So they put more steps in the mechanism and, like I said, demonstrated it in the lab. So that's really cool. So again, we're looking at the Discovery Institute claiming, oh, you know, this stuff doesn't count. What you need are new proteins with new structures. Turns out we have those both in the lab and in nature, new proteins with new structures that didn't exist before. We've documented the thing that they're asking for. So just to frame this a little bit, they moved the goalpost to somewhere where they thought we didn't have the answer. But it turns out we have the answer for the new location of the goalpost as well, which I think is cool. So let me put this screen back. We'll go back to the article. So let me remove this. Let me go back to the article. Hopefully I don't mess up the screen sharing at any point during this. Where's the article? Help if I pulled that up. There it is. There we go. Share that again. And we're back. Great. Okay, so I can get rid of this now. And we're back with the article. Okay. Oh, I should probably add the article back to the stream. There it is. Okay. Whew. So there's a lot going on in this claim, and it's not great. It's not great for the creationists here. But let's keep going. Let's keep going. My feeling is that creationists are going to reject most of what I just talked about, because with the exception of that last thing, the yeast example, with the exception of that, all of those were not directly documented in the lab. They were done, you're looking at genomics and things and doing comparative genomics to find them. But I think that, well, you're just inferring it based on nature. It wasn't done in the lab. It wasn't directly observed. I think that's a self-defeating argument for intelligent design proponents, unless they want to start demonstrating the mechanism of intelligent design in the lab, which I don't think anybody is prepared to do. So I'm going to leave that there. So let's continue. We are almost done already here. So we're going to go down here. This is the last paragraph. Wrap it up with this bit, the challenge for evolution. And this is kind of interesting, I think. So he says, the central challenge for evolutionary theory is creating sufficient information to produce something truly novel that functions at a level that would benefit an organism. He then goes on to say, and he's talking about here, talks a little bit about the lac operon. In the case of the lac operon, the specificity required for it to function is not the difficulty of an RNA polymerase binding to the promoter region. I mean, that is a specific sequence. That is specificity. Maybe that's not hard to get, apparently, because we showed experimentally it's easy to get, but that is specified information that is required for it to work. But this is the key part right here. The specificity and thus the information reside in the sequences that encode the repressor that acts as an on-off switch in the genes that encode the proteins that break down lactose. They're moving the goalposts here by responding to a completely different argument than the one I made, because now they're not talking about the regulation, right? Not talking about, that's what the promoter is. The promoter is a regulatory region, right? It's the transcription initiation site. Mutate it, you can block it, right? It's regulatory. So that's not what they're talking about here. Right here, they're talking about specific genes, the repressor gene, that's a gene outside of the lac operon that affects lac operon expression, and then the 3 genes within the operon that are required to break down lactose. This is a different argument than the one I'm making. They're specifically talking about protein coding genes. I didn't mention protein coding genes. You're responding to me. I mentioned random sequences, some of which were protein for peptide encoding, some of which were long non-coding RNAs, and an example with the lac promoter. I don't know why they're bringing up protein coding genes here. That wasn't part of my video specifically. I could argue that novel genes can evolve, right? De novo protein coding genes are a thing. I could make that argument. I'd be happy to. I've provided several examples. Just earlier, I provided several examples. I could argue that novel regulatory schemes can evolve. I could totally do that. We saw that happen in the Lenski long-term evolution experiment with the CIT+ lineage. That was a novel regulatory scheme. We also see this in humans with lactase persistence, right? Humans that are able to digest lactose throughout their lives. That is a change to the regulatory scheme of the lactase gene in humans. Contrary to what creationists claim, it's not just a broken repressor. It does actually, you're upregulating it with an enhancer in the most common forms of it. I think I've talked about that elsewhere. That's an example of a new regulatory scheme, right? I could make those arguments, and I would be happy to, but I did not make those arguments in the video that Discovery Institute is responding to here. So in summary, Dr. Stephen Meyer from Discovery Institute made an extremely specific claim. His claim was incorrect, which I pointed out, and I provided some very specific counterexamples explaining and demonstrating how and why his claim was incorrect. Discovery Institute responded without really addressing what I actually said. They made a different argument, right? What they did was, in effect, concede the argument I actually made without saying so. They conceded that point, and then moved the goalpost to a higher standard of information, right? Ooh, new protein coding genes with new structures, right? Now that position, it turns out, is also invalid. I could go on for much longer than here tonight, but we don't need to. I showed several examples. The position they staked out of de novo protein coding genes, also invalid. But my proximate point that I started with stands. You don't need an intelligent force of some kind to generate new biological information. You can do that via undirected processes, right? And that hasn't changed. So before I go, I just want to point out a few things, just a few kind of closing ideas. Step back from this specific paper, this specific post, and kind of talk big picture here. First, I want to just briefly kind of point out how I make my arguments. right? In my creation myths series, the arguments that I make are extremely specific and extremely narrow. So I take very particular, specific creationist claims and I address them. And I always show the creationist, either in text or in video, making those claims. Or if I don't show it directly, I'll say, like, here's the book. You can go to the book and find the creationist making this argument, right? And I respond very specifically to the argument that I'm, focusing on. I do that on purpose. I don't want to get into sprawling, is evolution true arguments. That's pointless. It's a giant waste of time. It's too broad. There's never any conclusion to that kind of discourse. I want to break things down into tiny questions that can be addressed quickly and simply. Is evolution true? Pointless. It's going to just spin off in a million directions, and you'll never actually finish a thought, right? You'll never finish an idea. Can non-intelligent processes generate new biological information? That's a question we can address very quickly. The answer is yes, and here's how we know, right? So that's what I'm doing here. Now contrast that to what the creationist approach to this kind of thing is. This is, I'm using this as an example, but this is an extremely common thing. This is the second thing I want to point out here, is that the strategy is to ignore the narrow point, right? Pretend the argument is about a broader point and then argue that broader point. In some cases, this is very extreme, where whatever the narrow point is, it's kind of whatever buzzwords are in there, it just like slots you directly into a script. And then like the script is just going to be the same each of the 5 times somebody talks about a specific thing, right? That's something you see. But more generally, it's basically you ignore the narrow point. If you don't think you can address that narrow point sufficiently, you move to a broader point, right? Move the goalpost to something that the creationists think hasn't been demonstrated. Or they can argue, oh, well, it hasn't been demonstrated in the lab. You're just inferring with historical science or whatever, right? Now, of course, there's a double standard here, right? Because if it's outside the lab, it's not directly observed. It's historical science. We've all heard that. It's not operational science, yada, yada, yada. But if you do it in the lab, what do creationists say? Oh, that doesn't count. That experiment was designed by the experimenters. That doesn't count as undirected. They designed that experiment. That's how you got those random sequences to generate the function, right? So there's a double standard here, a little bit of heads I win, tails you lose kind of thing going on. But put that aside, the inevitable conclusion of this tactic is origins or bust, as my friend RJ Downard likes to describe it, right? Basically, we're going to play whack-a-mole until we get to the ultimate origins of biological information, all the way down the causal line until we get to abiogenesis. And then like 90% of the time, this kind of thing ends up at an abiogenesis argument. You start with something like, irreducible complexity, or you can't get biological information, and you show that that actually is the case. Oh, well, how do you get the genes on the lac operon? Oh, here's how you get new de novo genes. Oh, but how do you get all the interactions in the genome? Oh, well, here's how, regulatory networks can evolve, and here's how, the homology between different. Oh, well, how do you get the cell in the first place? That's way too complicated. You end up at abiogenesis every time, right? With this kind of argument. So that's kind of where I want to leave this. The last thing I want to show you, I'm gonna take this screen down, and I'm gonna share one more thing, because I found this today on Twitter, and the thing that I want to leave you with is the tweet from Dr. Stephen Meyer, where he linked to this on Twitter, because this is just perfect. So I will share one more thing with you. Here is the tweet that he sent out linking to this article, and this is how I found it this afternoon. He said, in short, the sequences performed no new function, so no new information was created. And that tweet is just the perfect encapsulation of the game that's being played here, of the degree to which we're going to move the goalposts away from the specific thing that I was talking about, right? They're, in theory, responding to a specific thing that I said, but this has nothing to do with it. You get sequences that did a function, did a specific known function in one case. They documented a specific function, but it wasn't a new function. That's the new standard. So no new information was created. And that, my friends, is a world-class goalpost move in response to an argument for which they have no good counter argument. So that is my response to my friends at Discovery Institute taking issue with my creation myth that you need an intelligent source to generate biological information. No, you don't. I demonstrated why, and they did not have any counter to that in terms of addressing my argument. Thank you all for being here for this little journey with me. I know it's on short notice tonight, so I appreciate you all being here watching. I see 41 people watching, 23 people liked. Come on, hit that like button before we end this. This is fun. Or hit the dislike button. That's fine too. You know, if I make you angry, that's fine. Anyway, thanks everybody. I appreciate you all being here. Just a quick programming note. I am recording this on Tuesday night, the 26th of April, and I am having on Thursday afternoon, the 28th of April, I will be chatting with Dr. Herman Mays. We are going to be talking about traced and Dr. Nathaniel Jeanson's methodology. That is going to be fun. If you see this before 2 p.m. Eastern Daylight Time on April the 28th, 2022, you're going to want to see that. It's going to be a lot of fun. If you're watching this after that time, or if it got postponed or whatever, you're still going to want to watch that. Just, have to find it on your own. So thank you for watching, everybody. Have a great night. I will see you, I hope, on Thursday afternoon.